Yes, my name is Sergey Yaknin. I'm the Chief Operating Officer at SOPHiA GENETICS. I'm here because today is a very exciting day for many cancer researchers around the world. Today is the day of the publication of the papers from the Pan-Cancer Analysis of Whole Genomes project, which was world's largest cancer genomics project that has been going on for about 6 years. It's really the culmination of about 6 years of work of research institutions from around the world trying to put together their resources to analyze the DNA of cancer patients collected all around the world at a national level and trying to detect patterns of mutation that may be acting across different cancer types. My role in this project was to lead the technical working group where our challenge was to collect the data from all of these different national level cancer projects, deposit it into a data repository, really a series of data repositories around the world, and then perform unified analysis of that data in order to be able to detect mutations, to annotate these mutations, and then to pass this data along to further downstream research groups in order to be able to perform their analysis. So this was really a hard challenge for us to solve. We were talking about collecting 725 terabytes of data, mostly processed in different ways by different institutions, unifying that processing protocol, and then deploying that data to cloud computing environments around the world and running analysis software on them to detect mutations and perform further analysis. This was really hard for us to be able to do, and it took us several years in order to be able to do it. Right, so Butler actually was a tool that I developed when I moved on from the technical working group into one of the individual analysis groups called the germline working group. The germline analysis was tasked with analyzing the germline genomes of cancer patients to try to understand the mutational processes that would have occurred there. In germline analysis, we actually see orders of magnitude more mutations than we see in the somatic genomes, and so this really poses even a harder computational challenge for us. And what I saw was that we don't really have any tools available in the scientific community now in order to be able to effectively do this. I brought to the job my experience working in the software engineering industry for many years before, most recently having worked at Amazon.com, where we ran large-scale software payloads on warehouses distributed around the world. And so one of the key lessons I learned from this was that when you operate at extremely large scale, you're going to see repeated failures occur, and it is what you do with these failures that really determines how effective you're going to be able to be. And so this is what I tried to do with Butler. Butler is an open source framework that takes other large-scale open source established projects and integrates them together into this comprehensive package that allows you to deploy a computing cluster onto virtually any cloud computing environment: AWS, Google Compute Platform, Microsoft Azure, academic clouds running OpenStack. You can very rapidly set up a computing cluster, set up the networking, set up securities so that you can securely do genome analysis on it, and then deploy bioinformatics algorithms in order to be able to do your computation on it. And what sets Butler apart from other tools is really this emphasis on operational excellence. How are things actually going to run in production? It has these comprehensive monitoring capabilities where I'm really obsessively monitoring everything that goes on in the system: CPU, memory, network, disk. How are the different processes interacting? That data is scraped off of the individual virtual machines and is aggregated into a time series database. That data can then be surfaced to the user in a series of dashboards using an open source tool called Grafana, but it can also be used for trying to detect anomalies, patterns when something goes wrong in the infrastructure, and take remedial action. And so this is what we call self-healing. There's a rules engine that is sitting on top of this time series database that defines what does it mean for the system to operate well. And when it detects that the system is not operating well, it can then take remedial action. It can boot back servers that have gone down, it can restart processes, and then all it needs to do is to send the human a message that says, hey, I detected this thing went wrong while you were sleeping overnight, I fixed it, the outage lasted 5 minutes, and so instead of coming to work with a crashed system in the morning as you normally would, you just just come back to a message that says, hey, everything's fine, and things were chugging along all night long except for this 5-minute period. And so this is what really allowed us to succeed rapidly in the germline working group and perform our analysis in a matter of weeks, whereas our initial analysis in the technical working group of the consortium took us years to complete. And so I think Butler in that sense really provided that extra oomph that we needed to be able to get the project done faster, and so I'm quite proud of it for that. What Butler really allowed us to do was to take all of the great tools that have been developed by the open source community, take them together, and using those accelerate the pace at which we do research for the benefit of cancer patients worldwide.